Microarray are mainly used for in order to get those transcriptional profiles where we are checking the gene expression. But in addition to that, these microarrays they are coupled with other techniques, and in this way we can have some other uses or multiple uses of these microarrays. For instance, it can be used along with chip that is called as chromatin immunoprecipitation. So what we do over here is we take those DNA segments which are attached to some proteins. We take those proteins and we design antibodies against them. So antibodies capture the proteins which are attached to the DNA. So in this way we can capture the DNA. So this works best with the whole genome telling arrays. If you want to look at it, we can have those transcription factor binding. In this case, you can have transcription factors which are binding to specific locations or motifs present in the DNA. So if you capture those, or if you get those transcription factors attached to those DNA, you can get those DNA and you can have an idea about their sequences. We can also have the idea about those post-translational modifications which are commonly known as PTMs, which are modifications like acetyl groups, methyl groups, etc. They are on the histone proteins. So here is the technique, that is chip. First chip is chromatin immunoprecipitation. The other one is obviously those the chip, microarray chip. So we have some proteins attached to the DNA. We capture them with the help of some antibodies. Antibodies are attached to a matrix. So antibodies, they capture these proteins. So then those proteins are since attached to the DNA. You get that DNA. Now you take that DNA and you can put it as a probe on some slides. You can get those fluorescently tagged or fluorescently labeled sequences. You already know them, so you can take those labeled sequences and use these probes which are actually you obtained from those target DNAs, and then you take those fluorescently labeled these single stranded DNAs and you let them interact with one another, hybridize with one another on a microarray slide. We get those labeled fragments, let them hybridize. So in this way we can get the color patterns in some areas where we can have detected their hybridization. We actually have detected their sequences. RIP is for RNA binding proteins, so it's kind of RNA immunoprecipitation. So that is similar to chip, but it is for discovering the RNA binding proteins. So rest of the things are the same as what we have seen in the chip. Chromatin or comparative genome hybridization, CGH. So that is ACGH, array CGH, comparative hybridization. So which is there to look into those cytogenetic abnormalities. So we can also design arrays for that. We take those DNAs, genomic DNAs, and we hybridize them to these arrays. And sometimes we use large clones, for example BACs, etc. As array features. In tiled arrays, we cover the genomic regions or whole genomes at a high coverage. So that is kind of we have a large number of tiles or small segments covering the whole genomes, so we can capture them or we can get them, and in this way we can have the idea about the overall sequence of that whole genomic region. So probes are used, they are designed to cover the whole sequence, usually excluding only simple sequence repeats, and there is no biasness here towards the non-transcribed region. So you take the whole genome and then you make those probes from that. So here is the genomic sequence you try to cover with the probes on array, and those probes are there on the array, and then you take your sequences and hybridize with them and see how much proportion of this genome has been conserved or how much proportion of this genome has been variable. In protein binding microarrays, we call them as PBMs. Protein binding microarrays can be used to identify transcription factor binding sequences or motifs. We take double-stranded DNA probes used on the array, purify protein hybridized to the array, and detect those proteins by the help of antibodies. And then we can use genomic sequences, or we can design oligonucleotides for that. So we can look at all possible 10 MERS nucleotide sequences on a single array. So in this way we can have the idea about those motifs or those regions with which those protein binding takes place, or protein molecules they bind. We see here in this section that these microarray techniques, which is actually a technique to get those gene expression, but with a combination of other techniques, it can be used for multiple purposes.